Patrick Hohenberger. I'll be talking about stem cells and stem cell research today. One of the most controversial topics in today's society is that of stem cells and the research involved with them. For those who do not know, stem cells are unspecialized cells capable of renewing themselves through cell division, sometimes after long periods of inactivity. Also, under certain experimental conditions, they can be induced to become tissue or organ-specific cells with special functions. Stem cells are important for living organisms for many reasons. In the 3 to 5 day old embryo, the inner cells give rise to the entire body of the organism, including all the many specialized cell types and organs such as the heart, lung, skin, sperm, eggs, and other tissue. In some adult tissues, such as bone marrow, muscle, and brain, few numbers of adult stem cells generate replacement for cells that are lost through normal wear and tear, injury, or disease. Given their unique regenerative abilities, stem cells offer new potentials for treating diseases such as diabetes and heart disease. Stem cells differ from other kinds of cells in the body. All stem cells, regardless of their source, have three general properties. Stem cells are capable of dividing and renewing themselves for long periods. They are unspecialized, and they can give rise to specialized cell types. Unlike muscle cells, blood cells, or nerve cells, which do not normally replicate themselves, stem cells may replicate many times. Our starting population of stem cells that replicate for many months in the laboratory can yield millions of cells. If the resulting cells continue to be unspecialized, like the parent stem cells, the cells are said to be capable of long-term self-renewal. One of the fundamental properties of a stem cell is that it does not have any tissue-specific structures that allow it to perform specialized functions. However, unspecialized stem cells can give rise to specialized cells, including heart muscle cells, blood cells, or nerve cells. When unspecialized stem cells give rise to specialized cells, the process is called differentiation. While differentiating, the cells usually go through several stages, becoming more specialized at each step. There are two different kinds of stem cells, called embryonic stem cells and adult stem cells. Both these kinds of stem cells have quite a bit in common, as well as many differences. Embryonic stem cells are derived from embryos. Most embryonic stem cells are derived from embryos that develop from eggs that have been fertilized in vitro in an in vitro fertilization clinic, and then donated for research purposes with informed consent of the donors. They are not derived from eggs fertilized in a woman's body. An adult stem cell is thought to be an undifferentiated cell found among differentiated cells in a tissue or organ that can renew itself and can differentiate to yield some or all of the major specialized cell types of the tissue or organ. The primary roles of adult stem cells in a living organism are to maintain and repair the tissue in which they are found. Unlike embryonic stem cells, which are defined by their origin, the origin of adult stem cells in some mature tissues is still under investigation. Human embryonic and adult stem cells each have advantages and disadvantages regarding potential use for cell-based regenerative therapies. One major difference between adult and embryonic stem cells is their different abilities in the number and type of differentiated cell types they can become. Embryonic stem cells can become all cell types of the body because they are pluripotent. Adult stem cells are thought to be limited to differentiating into different cell types of their tissue or organ. Embryonic stem cells can be grown relatively easy in culture. Adult stem cells are rare in mature tissue, so isolating these cells from an adult tissue is challenging, and methods to expand their numbers in cell culture have not yet been worked out. We don't know yet whether tissues derived from embryonic stem cells would cause transplant rejection, since the first phase 1 clinical trials testing the safety of cells has only recently been approved by the United States Food and Drug Administration, the FDA. Adult stem cells and tissues derived from them are currently believed less likely to initiate rejection after transplant. The use of adult stem cells and tissues derived from the patient's own adult stem cells would mean that the cells are less likely to be rejected by the immune system. There are many ways in which human stem cells can be used in research in the clinic. Studies of human Human embryonic stem cells will yield information about the complex events that occur during human development. The primary goal of stem cell research work is to identify how undifferentiated stem cells become the differentiated cells that form the tissues and organs. Scientists know that turning genes on and off is essential to this process. Some of the most serious medical conditions, such as cancer and birth defects, are due to abnormal cell division and differentiation. A more complete understanding of the genetic and molecular controls of these processes may yield information about how such diseases arise and suggest new strategies for therapy. Human stem cells could also be used to test new drugs. For example, new medications could be tested for safety on differentiated cells generated from human pluripotent cell lines. The availability of pluripotent stem cells would allow drug testing in a wider range of stem cells. However, to screen drugs effectively, the conditions must be identical when comparing different drugs. Therefore, scientists will have to be able to precisely control the differentiation of stem cells into the specific cell type on which drugs will be tested. Perhaps the most important potential application of human stem cells is the generation of cells and tissues that could be used for cell-based therapies. Today, donated organs and tissues are often used to replace alien or destroyed tissues, but the need for transplantable tissues and organs far outweighs the available supply. Stem cells directed to differentiate into specific cell types offer the possibility of a renewable source of replacement cells and tissues to treat diseases including Alzheimer's, spinal cord injury, stroke, burns, heart disease, and diabetes. To summarize, stem cells offer exciting promise for future therapies, but significant technical hurdles remain that will only be overcome through years of intensive research. So despite the controversy behind this topic, the potential of medical advances that could be made with the use of stem cells is in great supply. Thank you.